Recording now?
Mm-hmm.
All right. So hello everyone and welcome to today's UVic Center for Biomedical Research Café Scientifique. And I'm just going to mute some of the notifications, but not the presentation. So I'm Dr. Leanne Swain. I'm an associate professor in the Division of Medical Sciences here at UVic. Virtually. And I'll be your host and question moderator tonight. So I'll start by acknowledging with respect the Lekwungen peoples on whose traditional territory the University of Victoria stands, and the Songhees, Esquimalt, and Wasanich peoples whose historical relationships with the land continue to this day. So if there's anyone in our crowd tonight who's new to Café Scientifique, what we do is we welcome scientific researchers affiliated with UVic to share their work with the community. So pre-COVID, as you know, as many of you know, this was in person, and so for the time being, we're doing this virtually. So we won't have any interruptions for questions during the talk, so please just note your questions down or post them in the chat, and then I will read these out later to the speaker at the end of the presentation. So the talk is going to run for about 45 minutes, and I'm super excited for today's talk. So we have with us today Dr. Hosna Jabbari as our speaker, and she's an assistant professor of computational biology in the Department of Computer Science. So she heads up Team COBRA, which I think is a really badass acronym for Computational Biology Research and Analytics, and I'm going to pass it over to her. So welcome, Dr. Jabbari.
Thank you, Leanne. So today I'm going to present some of the efforts that our lab has done already on COVID, what we know about it, what we thought we knew, and what we think is going to happen in the future, and what the next steps are going to be. So we are all familiar with what is COVID-19. We know that it's caused by the SARS-CoV-2 virus, and it has already infected over 48 million people worldwide. It has caused more than 1.2 million deaths, which is awful. And it's believed to have emerged from a bat-borne virus. This virus is a positive sense single-stranded RNA molecule, and this is what we should all know by now, that it has been disrupting our life for the past 10 months. While it has been an awful case as a pandemic and has affected our life in many different aspects, it has created a collaborative environment, bringing together academics and industry partners from all over the world to find a solution to end this pandemic. We joined this efforts of collaborative research for ending this pandemic in March, and today I'm going to show some of the results that we have had so far. This is a picture of me and my students, our badass COBRA team, and we are mainly focusing on curing human diseases by developing novel diagnostics and therapeutics. And these efforts are using techniques from bioinformatics, machine learning, data science, and computational genomics. Our expertise focuses on RNA interaction, RNA structure, transcriptomics, and working through RNA interaction with proteins and RNA therapy. So this virus, being a positive sense RNA virus, was an opportunity for us to take a look and see if we can apply our expertise to understand this virus a little bit better. What do we know about this COVID-19 disease? We know that the current diagnostics are using QRT PCR. We know that some of these patients, people that have been infected by the virus, show no symptoms. The hypothesis about how this virus enters the cell is through the ACE2 protein. And we know that the symptoms that patients have are varied. Some of them are very common, like fever, cough, and chills, which is similar to cold and flu. Some of them are more rare and more probably troublesome, like the COVID toes, heart conditions, and brain fog or delirium. The picture on the right shows an example of this COVID toes, which is the coagulation that you see on the finger, not on a toe, and that's my finger in January 2020. Some of these symptoms are more severe in the elderly, and we don't know what other symptoms or side effects these COVID-19 is going to have. So what we don't know are some of these questions. We don't know why we have different symptoms in different populations. We don't know why some patients are asymptomatic. We don't know if there are going to be any long-term effects in different populations. Are there going to be long-term effects in general, or are there going to be long-term effects in kids or adults? We don't know if some of these effects are going to be causing chronic diseases like heart condition or Alzheimer's. And we don't know what type of mutations we're going to see on this virus and what type of effect these viral mutations are going to have. So in our team, we tried to look at COVID-19 and this SARS-CoV-2 from different angles. We tried to see if we can find any better test. Can we find a computational framework to analyze the sensitivity of the existing diagnostic tools? Can we use that to better design efficient aptamer-based tests so they don't need the QRT PCR anymore? We looked at possible treatment options. Are there any areas of the virus that we can focus for a vaccine or for a treatment option to lessen the severity of the symptoms? For that, we looked at mechanism of function of the virus. Some of the parts that we look at was a very hot area of the virus that is called the frame-shifting, ribosomal frame-shifting pseudonot. And we have some results on that I will be presenting today. We also are looking at what are the areas of this RNA virus that are causing thrombosis or coagulation. And we are looking to see if we can identify the areas that are causing delirium, brain fog, and their possible long-term effects. So 2 projects are already highlighted. These 2 are the ones that are almost complete, and they are available on bioRxiv. And I will be focusing mainly on these 2 projects for this talk today. I try to keep everything very informal, but if there is anything that doesn't make sense, please make sure to ask, and I will be more than happy to explain more. The first project is in silico prediction of COVID-19 test efficiency with a program we called Dyno Knot. Dyno Knot is a name we chose for the duplex interaction of nucleic acid with possible Pseudonot. And the project was led by Tara Newman, my undergraduate student who did her USRA with me over the summer, and by Kevin Chang, who was my undergraduate student at the University of Alberta. The goal of the project was to predict how the structure of the SARS-CoV-2 virus affects primers in binding to their target. So practically, we wanted to find if to create a computational framework that can evaluate binding capabilities of the current primers that are currently in use for testing. You see on this slide animation of the portion of SARS-CoV-2. These SARS-CoV-2 has many different parts that are identified by different areas of the gene. And you see some areas, the blue areas, that identify some of the target areas of some of the current tests. So while some of these tests, like number 1, number 2, and number 3, each one of them target a different area, tests 5 to 9 identify similar areas. And the work that we did There was some experimental work on the efficiency of these current QRT PCR test sets, primer probe sets for COVID-19 that at a time was a bioarchive paper, and now it's been published in Nature. That's the Vogels et al. Paper that I will be referring to throughout the talk. We chose to work on the same sets, not because they were just available, but because they were experimentally validated, and we could kind of validate our computational findings as well. So let's see, how does QRT PCR work? We have, at the beginning, we have the positive sense RNA genome that you see in purple on the slide. And we have a reverse primer that is complementary to a target area of that genome. Reverse primer binds to this RNA. Reverse primer is of type DNA molecule. So binds to this RNA, and then the reverse transcriptase creates the negative sense complementary DNA. When we have the negative sense cDNA, then the forward primer binds to that, and DNA polymerase creates the positive sense cDNA from that. And then again, reverse primer attaches to the positive sense cDNA, and this process repeats many times, so we have enough of the virus for tests. As you can see in this slide, we have, in the first case, we have a DNA-RNA interaction. In the second case, we have a DNA-DNA interaction, and last one with a positive sense cDNA and reverse primer, we have also the DNA-DNA interaction as well. So this reverse primer is a DNA and can interact with the RNA and the DNA molecule. You may be asking that the reverse primer is complementary to this RNA or DNA, so why should we at all think about the structure of this molecule? In this slide, we have the reverse primer on the left side. It's a 26 base molecule that's from the RDRP primer probe set. And on the right, you see a portion of the gene for this, the target area for this reverse primer. And as you can see, this is the secondary structure, the two-dimensional structure of the gene. Not all bases are equally available for the primer to bind to them. So you can't just say because the primer is complementary to the target area, it is going to bind to its target area, because it has to compete with the rest of available molecules in the environment for binding. So structure can have a big effect on this. But to look at the structure a little bit better, let's introduce another way to take a look at this structure. On this slide, you have a simple, small secondary structure. In this secondary structure, this is an RNA molecule, because we have the A, C, G, and U bases. These RNA molecules are single-stranded, so they have two distinct ends that, as you may have noticed in the previous slide, they're called 5' and 3' end. And you see that the base pairs on this slide are shown with the lines connecting the two bases, like UA, CG, and AU. These bases are complementary and they bind together. A normally binds with a U and C binds to a G. So the two ends of this molecule are these A and U. If I just take the two ends and try to pull apart the ends, so the backbone of the RNA, which is the line that shapes all the sequence, becomes a line, while still the base pairs are available and the base pairs are not disrupted, I can see a figure that we call it an arc diagram. It's going to look like this. So bases 1 to 55 are the sequence, the RNA sequence that we have, and the arcs represent the binding of these bases. As you notice, this UA is this one, and the end, that AU at the end, is this base pair. So one characteristic of this structure is that all the base pairing happen in nested shape. So you can see that bases are like rainbows. They are just one inside another one. But all are the structures like this? I can say no, because apparently the question was leading to that. In the second structure that you see over in this slide, we have another RNA molecule. In this RNA molecule, we have unpaired bases of one loop, base pairing with unpaired bases of another loop. This is what we call a pseudonotted structure. And if I do exactly the same thing, pull apart the two ends of the molecule and present that in an arc diagram, I'm going to see a structure like this one. In this structure, in addition to the rainbow nested base pairs that I would see, I have these crossing base pairs. These two crossing base pairs that you see over here correspond to the two long-range interactions in green in this slide. Crossing base pairs are a very like the main characteristics of pseudonotted structures, and amazing enough, prediction of the structures that consider pseudonotted base pair, crossing base pairs like this, is very, very difficult. So what we did for the tests that are currently available was that we looked at the structures of the area that the target area together with the reverse primer. So on this slide, you can see that the target area is the big area you see on here, and the target area for binding of the primer was shown in green. And some of this it's very small, I understand, and the reverse primer is shown with red. So we developed a program called Dyno Knot, and this Dyno Knot considers the structure of the target area and the primer before their interaction, and then decides if they can possibly interact together. Like binding the two of them will be beneficial enough so that they leave their previous binding partners to attach to one another. And this is what we have tested over here. As you can see, we have the 9 cases. In some of these, you see repeat in the names, like these two and the bottom two. I will explain that later. But as you can see, some of them have very strong bindings, like all the reverse primer binds to the exact target area. In some others, we don't see a great interaction. If you look at this one, for example, the HKU and R, this is the primer and this is the target area. You can see that primer is binding to itself and the target area is binding to other bases around it. This structure was created at 37 degrees. So this is the structure of the molecule that we predict would exist at 37 degrees. But while we have QRT PCR, we go through the annealing process, and then we have a higher temperature. So we decided to take a look at this to see what happens if we have higher temperature. On the left side, you see the interaction in higher temperature, and this is 95 degrees. And in 95 degrees, you see almost complete interaction between the primer and the target area. We did the same thing with the other one. That's another primer probe set that we will see later on the slides. And on the right, you have it at 37 degrees. We don't have a perfect binding. On the left side, we have it at 95 degrees. And you see almost perfect binding over here. So let's zoom in on the primer probe set that we discussed. As you see over here in the 37 degree, we have the primer interacting with itself. The target area was interacting with itself. At 95 degrees, the primer is almost completely interacting with the target area, while still some of the base pairs are pairing with the area that is not part of the target. The same with the target area. Some of the bases at target are interacting with other parts. But the question is this going to be strong enough binding for the primer to attach? So we noticed that in one of the primer probe set, we have degenerate bases. one of the degenerate bases was an R and the other one was an S. The R base can be a G or an A, and S can be a C or a G. And when we have the degenerate bases in there, that means we have a mixture of all these different primers and probe sets. So we decided to analyze the efficiency of each individual ones instead of looking at them as a pool. Here we have the 4 cases that you can get from R being a G and S being R being A or G and S being C or G. And in the Vogel's paper, the one that looked at the efficiency of these primer probe sets experimentally, they suggested that maybe if the degenerate bases were included A, that would be a better binding. So we tried that case as well. So the case E and F are when R is an A and S is an A, and R is a G and S is an A. So S was assigned an A instead of C or G. If we focus on some of these, you will see that in some cases, like when S is a G and R is a G, we don't have a good binding at all over here. In some other cases, and the same when we have R is an A and S is an A. In general, when S is an A, we don't have a good binding, so we don't agree with Vogel's at all about the binding of the S. The good point is that their suggestion was a hypothetical suggestion. They didn't actually evaluate that experimentally. So we believe that this is not going to make this primer probe set more efficient. Looking at the partial mismatches among the primer probe sets, we saw that mismatch was seen in some of these primer probe sets. These 4 of the primers had mismatches. In ENCOV and Sarbeco HKU primer probe set had analytical efficiency and sensitivity similar to the others. That was experimental analysis done by Vogel et al. When we looked at the ENCOV RN2 reverse primer, we saw a single base mismatch, and we think that would be unlikely to affect the binding of the primer and probe set. And in the experimental result, they show high analytical efficiency and sensitivity. In Sarbeco and HKU, we saw mismatch at 37 degrees, but we didn't see a lot of mismatch at 95 degrees. We believe that should also be fine with the current setting because that's a QRT PCR, but we don't suggest using them in lower temperatures. In the RD-RP primer sets, we had degenerate bases, and we identified that the best performing primer probe sets are when R is assigned a base A and S is assigned a base G. Then the other question that we wanted to answer with the current existing primer probe sets was how much they are going to be affected by mutation. So on this slide, we have a scheme of the RNA genome of the virus, and up top we have the mutation diversity among all possible bases. I understand that this is a very small figure, but I will have a bigger one in the next slide. So in this slide, we have all these primary probe sets. The target areas is marked with a marker over here, and on top of that, we have the mutation rates or mutation frequencies in a specific area. Focusing on one of them, this is the HKU primer probe set. You see that the target area for these primer probe set is an area that has a lot of mutations around it. So we decided to take a look at possible mutations and their effect on binding of the primer. We looked at all the mutation, all possible cases of mutation at the reported mutation positions. And over here we have the G mutated to an A and at different positions and so on. And we looked at the different types of binding that can happen with the mutations. Over here we have the small figures. As you can see, in some of the mutations, there is possibly no change. In some other mutations, you can see there is a big change in the target area, not binding at all with, in the primer area that is not binding to its target. So we looked at the effect of mutations. We found that in 3 of the primer probe sets, mutations were very effective. In E-Sarbeco primer mutation, we saw a complete mismatch with the reverse primer and RNA binding, as well as reverse primer and cDNA binding. This can be very effective and can possibly cause problem with detection. In the HKU primer, we saw mutation at position 29,148, while the forward primer was not able to bind. We believe there's not going to be any amplification because of that. And at the NCOV N3 primer, we saw a complete mismatch with the reverse primer with RNA binding, but reverse primer can still bind to the cDNA with one mismatch. We believe that may result in amplification problems, but we're not sure because the cDNA binding is still fine. So looking at what Dyno Knot, what we could get out of the Dyno Knot, we can summarize that about COVID with the current test. We see that the reverse primers, because we're using QRT PCR, we saw that the reverse primer can bind to their expected target and the viral structure wasn't preventive of this interaction. We saw low efficiency in the RDRP primer probe set. That was the degenerate probe set, primer probe set, and we identified the best performing one of the set. So we believe replacing the degenerate bases with the one that is performing better can result in higher efficiency. We predicted some disruptive mutations and their effects on the primer probe sets. So some of the mutations that are more prone to being disrupted for the primer probe sets should be avoided. And the other thing that we have noticed is that in the current setting with the QRT PCR, we need a high viral load for detection of the virus. So it's very sensitive about the replication process and binding of the primer to the target area. We believe if we have different kind of tests like aptamer tests, it can be more sensitive to lower viral loads. The aptamers would need to, aptamers are small molecules. They are similar to the primers. They are RNA-based molecule or DNA-based molecule, and they can, in normal temperatures, 37 degrees or room temperature, they can bind to their targets and they can be fluorescent and identify binding. Because the tests are more challenging in children, we might think that it might be caused by the viral loads in children, and we believe if we have more sensitive tests, that might have been a better answer for that. Now, for the second part of the talk, I would like to change gears into ribosomal frame shifting Pseudonot. This ribosomal frame shifting Pseudonot or ribosomal frame shifting is a key step in replication of coronaviruses. And in this slide, we have a figure of one area of the SARS-CoV-2 virus. And this area has 2 open reading frames, as you can see in this slide, ORF1A and ORF1B. These open reading frames overlap, and therefore, in a normal situation, only one of them should be translated. However, there is a pseudonoted structure at this overlap region that facilitates a frame shifting and translation of a fusion of these both ORF1A and 1B. This area of the RNA genome is a hot topic right now for therapeutics, because it has been shown before that for the previous coronaviruses, if we can disrupt this structure, the yield of reproduction will slow down. So that can be a case for reducing severity of the disease for patients with severe symptoms, for example. This work was done by two of my students, my PhD student Luke Trinity and my undergraduate student Lance Lansing. The goal was to identify structural similarities between the -1 programmed ribosomal frame-shifting pseudonots of some of the well-known coronaviruses like SARS, SARS-CoV-2, and MERS. So with the SARS, we know that the Pseudonot sequence is very much conserved. In SARS-CoV-2, the same sequence almost identically exists for that. So we have this 68 base pair sequence that is quite identical to the previous case SARS. We have seen from SARS that Pseudonot plasticity is very important for viral replication, which means that if there are structures that are equally stable but different in shape, that can transform from one structure to the other, then the Pseudonot has better success rate and can have a better yield when it's going through the replication process. So we wanted to look at Pseudonot plasticity for SARS-CoV-2 as well. We also have seen mutations in nearly every base of the SARS-CoV-2, and these mutations have been identified on the figure on this slide with red arrows. As you can see, we have 3 mutations that are near the junction of stems. One of them is the U20 up top over here. The other one is G29 over here, and U58 over here. And then one is adjacent to an adenine bulge. So if you look at this adenine bulge over here, we have this mutation. And then there are 2 mutations in the loop, the C and the U mutation. When we look at the mutation, the first one, the first character is the base that it is in the reference sequence. The number that comes after is the position based on this 68 base pair place or sequence, and the second character shows what this base is going to be mutated to. So a U20C says that this base U in the ref sequence would turn into a C in the mutated sequence, and so on for the rest of them. So to take a look at that, we went through a very complex process. The first thing we did was that we tried to align these sequences using a de novo alignment method. Using this de novo alignment, we don't do a sequence alignment. We do a structural alignment. We find base pairs that seem to be important in this alignment. That means if there is a mutation on one of the bases, you see a similar mutation on the base that is complementary to it, to the reference structure. That suggests that this base pair is structurally important, functionally important, and needs to be conserved. So after doing that, we identified an area that is marked with the stars over on the bottom row of this table. This is the 68 bases of the ribosomal frame-shifting Pseudonot. And this one, we have seen the sequence and structures over for the SARS-CoV, the first line, and SARS-CoV-2, and MERS. This, the consensus structure is the fourth line over here. The interesting part is that, yes, we expected the structures for SARS and SARS-CoV to be the same because they are very much similar. But something that was unexpected and experimentally not yet validated is the similarity that we are going to see between MERS and SARS-CoV-2. And the sequence of the structure that seems to be the case for these two, for the area of the Pseudonot, are pretty much conserved. So keep in mind that we don't have programs that do Pseudonot alignment, and these areas that we are looking at are all pseudonotted. That's why you will see some changes in the structures. The structures have been shown in two-dimensional in the bottom rows, and the red bases show the pseudonot part of these structures, the program ribosomal frame shifting pseudonot for all of these viruses. As you can see, none of these structures are pseudonotted because you don't see long-range interactions. But when these red sequence forms into a pseudonot, their pseudonots are going to be very similar in these red areas. So then we took another step further to see what type of structures we can find for the program's ribosomal frame shifting pseudonot, that these structures are similar in the energy but different in their shape. And that was something that we thought might inform us about the plasticity of the pseudonot, which seemed to be important for SARS at least. So what we did was that we followed this diagram. The first thing was, of course, we looked at the sequence. We found some energetically favorable initial stems. And then based on those stems, we tried to find, okay, if these initial stems form, then what type of a structure would form after. This follows a hypothesis called hierarchical hypothesis for RNA structure formation, which says that the full structure of the RNA doesn't form in one go, but first some base pairs pair together, some bases pair together, and when the structure is closer, when the sequence becomes closer to one another, like more compact, more complex base pairs also form, and these complex base pairs might be pseudonotted. So we followed the same hypothesis and we predicted the secondary structure for this sequences of these molecules. Let's first take a look at the initial stems that we identified for SARS-CoV-2. We have the sequence of the 68 base pair in the bottom, and these 18 cases that you see with dot bracket are a simplified scheme of the arc diagram. Each open bracket shows the start of a base pair, and the closest closed bracket shows the base that pairs with that one. So in this structure, these two base form, and as you can imagine with the arc diagram, these are all nested structures and there's no pseudonot in the original phase. So then we looked at these structures and we fed them to a program that we have designed in our lab called iterative H-fold, and we formed the final structure. Amazingly, out of the 18 initial structures, we could only get 12 distinct structures as output. As you can see in this slide, some of them, no matter from which one of these starting points you start, you will get the most stable structure to be this structure, and this structure is the structure that was reference structure identified as the reference structure in SARS, and is believed to be the reference structure for SARS-CoV-2 as well. Then you see that some other, with different starting point, you can get to the same end point, and that might mean that these structures are more likely to end up in. Then you see some structures, so these structures are all ranked based on their free energy, and you can see that they are similar structures that are highly likely to form, and they are not very different in terms of their free energy, which can hint to the plasticity of this pseudonotted frame shifting structure. Then we looked at possible mutations that can happen in SARS-CoV-2 and the effect of these mutations in the plasticity of the pseudonot. So over here, you see all the mutation, the 6 mutations that we saw in the first slide for program ribosomal frame shifting. In each case, we identify the initial structures. Over here, you will see the structures that are different from what we identified for the reference sequence of SARS-CoV-2. And we followed the same procedure for all of them. And this one, for example, is the U20C mutation. And in this case, we have a different endpoint, and this different endpoint seems to happen for more of the initial starting point. This is highly stable, different from the reference structure. And plasticity seems to still exist over here because we have a lot of structures close to one another in terms of free energy, and some of them are highly accessible. No matter which point of these initial sequences, initial structures you start with, you will end up with the output structure. So then we did the same thing for MERS. We started, so the first thing is for the MERS reference structure, and we started with the 19 identified original starting point, and then we looked at the structures. The structures, again, we had a lot of plasticity observed over here, and the similarity of the structures between what we got from MERS and SARS-CoV-2 in terms of the type of pseudonot was very interesting. So summarizing our results, we saw the structural similarities between SARS and SARS-CoV-2. It was very much expected because, as I mentioned, the 68 base of the pseudonot sequence is 99% identical. What was interesting was that we detected the structural similarity with MERS and SARS-CoV-2. It is interesting because there is almost no sequence similarity that you can detect between these two RNA viruses. In fact, if you do a blast with this sequence of the programmed ribosomal frame shifting for the two of them, you will not get a hit. You will not detect anything. This structural similarity was hypothesized by another bioarchive work in 2020. That's the that we referred over here, but it has not been experimentally validated. So maybe our new data will be a kind of another point for validation of this structure, and maybe someone, a physicist, will check this structure experimentally, and that would be lovely to see what happens. We also identified multiple of non-native structures that are energetically and structurally very comparable to this SARS-CoV-2. This suggests for the flexibility and conservation of the pseudonot in this area. And we identified some non-native structures that were very distinct from both SARS-CoV-2 and MERS. They existed many distinctly different structures in both of these frame shifting areas. And it would be interesting to see what is the effect of these molecules. They're not at all similar to the native structure for these pseudonots, but they are energetically close to these pseudonots. So it would be interesting to take a look at their possible function. Then we looked at mutations for these areas, and the structures that we saw out of the mutated sequences was quite different from the native structures. And in some of these mutations, for example, the U20C that we looked at the structure, it has been hypothesized that the pseudonot is conserved after the mutation. And we looked at the structure for this one. We saw that there was a significantly different structure from the native expectation for the structure, but had a low free energy. And that would be interesting to see if that structure would have any function in the conservation of the overall function of this pseudonot. Another student of mine, Allison, is looking at similarities similarities, the structural similarities of this SARS-CoV-2 with other viruses. These are different areas, different viruses that have their phylogeny on the left. And on the right side, you see a diagram of homologous and non-homologous areas of these viruses. The homologous ones are in white and non-homologous ones are in blue. So we are interested in both of these areas, homologous and non-homologous ones, because the ones that are similar can show us what they have in common, and the ones that are non-homologous can show us that the difference in function of these, then can identify differences in functions of these viruses. So if we go with the principle of inclusion exclusion, maybe we can identify some specific functions and functional structures in these molecules, and that's why we're looking at them. As you can see in some of these viruses, you see a lot of homologous sequences, while in some other ones, you see a lot of differences in the structure. And amazingly, in the 5' UTR, they are pretty much different in terms of the structure. So I hope that our results has inspired further research. We are very happy to collaborate with anybody who's interested in the topic, and we hope that with enough collaboration we can end this pandemic together as soon as possible. I will be more than happy to answer any questions you may have.
Great, thank you so much, Dr. Jabbari. That was such a great talk. And I was wondering if, I don't see any questions in the chat, but I'm just, so maybe just to get things started, I'm just wondering, in addition to sort of obviously affecting like diagnostics, and maybe you touched on this and I missed it, but what other like sort of information can you garner from this analysis of the RNA structure, like in terms of like just the virus itself, maybe like viral replication? Like, does it have any other impacts on the other properties of the virus?
Yeah, so there are a lot of things that you can get from the function, right? So we should remember that virus is a small molecule, and it needs so it has it's fighting for survival, right? So it can't carry baggage that it doesn't need. If it's keeping a part of its genome, then that might be very important functionally for the existence of the virus. So something that we are looking at, something that we are trying to find out of this inclusion-exclusion comparison, structural comparison with other viral sequences, is exactly like that. Can we find the structures that we can say are important for the potency or existence or replication of this virus? What happens if we can, and then what happens if we can disrupt these structures? Can we destroy the virus? Are we going to avoid any effects? Are we going to at least do something in terms of severity or the side effect of the virus, right? So these are all the things that we are looking at them from a functional view, a structural functional view, but it would be interesting to see if we can have other people validating our results as well.
Yeah, I guess that was my follow-up question, is like, how often do things that you're able to model, how often do those things, I mean, obviously there's a considerable, I guess, barrier to like validating them all experimentally, but like what are the barriers? And if you, I know that's probably a huge question, but if you can kind of speak to some of them, and then like, how do you prioritize sort of which pieces to validate, and what things are more easy to validate than others?
So previously, in previous collaborations that I've had, we had experimental partners, so partner that was interested in the same result. And for one of the areas that we have worked at, and they have a patent already for a similar project, we were looking at exon skipping for Duchenne muscular dystrophy. And we identified the best target area and possible structural effect on this one. And harnessing the result of our computation, they could design their antisense oligos, do the experimental analysis, and then that would reduce the number of experiments that they needed to do. So we kind of guided them. We never said that it's going to be, this is the absolute best case, but it can help in reducing the space that you have to search for the best result. So again, that's the same hope over here. If we can identify the area that we believe it's going to be to have the specific function, so instead of blindly looking for an area to just target for either testing or for the neutralizing this function of this virus, you can search around the area, or like if we identify a 68 case, exactly, narrow in and find the better, more optimal solution for that.
Nice. And so I guess I don't see any questions in the chat, but I guess another question sort of related is like, what's your next focus in this area? So what's, and just like, something that maybe gives us a little bit of hope, or what's your next kind of new goal, and what's your next project kind of related to this?
Yeah, so something that I think all of us are thinking is that, okay, we haven't been prepared for something like this. What can we do to avoid or move faster for the next possible pandemic, right? And I believe that we've been avoiding this kind of question for a very long time. If we had a computational framework to look and see, okay, this is my guide, I'm going to look for the solution like this one, other than just experimentally looking for it, maybe it would have been faster. It is actually quite nice to see that some of the RNA vaccines are performing much better than the other one. And these RNA-based vaccines were developed very fast because they were looking at some of this computational models and computational frameworks that they were using for designing these target areas. In Moderna, for example, I think they chose their candidate vaccines like in less than 2 weeks. This is amazing. This is what we should all be doing for all our experimental design, right? The whole hope is that what can we do? Can we make a system that can help us for the next possible case, to act faster, to design better vaccines, design better tests, be more prepared?
Yeah, very nice. Okay, well, that's really exciting, and just congratulations on a job well done. So how long have you been at UVic?
I joined UVic in 2019, so over a year now.
Yeah. Oh, exciting. Yeah, and you already have established this really dynamic and exciting team, and you're getting work out on bioarchives, so congratulations. That's awesome.
Thank you.
And yeah, it's really fascinating work, and it seems very, very challenging. So just excited that you're working on this issue. I don't see any questions from the group, but I think everyone's probably digesting the information. It's very exciting, very interesting work. And I'd just like to, on behalf of everyone, thank you for sharing this very important and innovative work with us. And I just look forward to seeing all of the exciting new stuff that's coming out from your lab. So maybe we'll have to follow up and have some conversations ourselves sometime.
Yeah, sure, definitely.
Okay, all right.
Thank you so much.
Yeah, thank you so much. Okay, thanks, Dr. Jabbari. Have a good one.
You too. Bye.
Bye.